ಓಕೆ ಓಕೆ ಸರಿ.
You know, smart. ಈಗ ಡೌನ್ಲೋಡ್ ಮಾಡ್ತಾರೆ. ಹಳೆ ಮೊಬೈಲ್
ಎಲ್ಲರೂ ಕರೆದ್ರು ಮೀಟಿಂಗ್ ಕ್ರೈಟೀರಿಯಾ 1 hour ಕರೀಲಿ. ಏನು ಆಫೀಸಲ್ಲಿ ಪ್ರಿನ್ಸಿಪಲ್ ಇಲ್ವಂತೆ?
Sir, very good morning, sir.
Good morning. Very good morning, all of you.
Yes, sir, you have joined. Just we'll wait for a minute, sir.
Okay, sir, no problem.
Yeah. And
Yeah.
we have made you as a co-host so that you can directly
Yes, sir.
share your screen, sir.
Yes, thank you. Thank you, sir.
Yeah.
Thank you. Yeah.
ನವ್ಯ,
ಬಾಳೆ ಸ್ವೀಟು
ತಿಂತದಾಳ್ ತಡಿ ಅವಳು
ನಿಧಾನ ಕೊಡಾಡ್ತದೆ. ಒಂದು ಅರ್ಧ ಸ್ವೀಟ್ ಇವಾಗ ಎಷ್ಟೊತ್ತು ಬೇಕಮ್ಮ ನಿನಗೆ? ಸೈನಿಂಗ್ ಮಾಡ್ಬೇಕಂತ ನಾನು ಐಡಿನೇ ಕೊಟ್ಟಿಲ್ಲ ಅದಕ್ಕೆ. ಸರಿ.
Vinod, sir.
Yes, sir.
Yes, sir. I think, can we start, sir, with your
Yes, sir.
permission?
Yeah.
Okay. So in fact, around 80 participants have already joined, and we have around 160 registrations, sir, which has occurred all
Okay,
over
sir.
Bangalore and Pan India.
Yes, sir.
Okay. Okay. Good morning, everyone. Participants, please mute once you join. So very good morning to all, and greetings from Saptagiri College of Engineering and Student Chapter, Indian Institute of Engineers, and Srishti Biotech Forum. Very happy to inform you all that this is a series of webinars which we are conducting from last 1.5 months, and we are we have done around 6 or 7 webinar series. And today, indeed, very privileged to have the most eminent scientist and Deputy Director of Forensic Science Laboratory, Government of Karnataka. We have with us Dr. Vinod Lakappan, sir. One sec, sir. I think it is, we are getting our YouTube echoing. One sec, sir.
Okay, sir.
YouTube echo. Sir, sorry for the disturbance. We had little bit of technical error with respect to YouTube live. We are on YouTube live streaming now, so it was little bit echoed. That's a reason. Okay, anyway, we are back now on stream. So we have around 90 participants who have joined, and as I was telling you people, we have Dr. Vinod Lakkapan sir, most eminent speaker, and we have Deputy Director. He is working as Deputy Director at Forensic Science Laboratory, Government of Karnataka, Bangalore. In fact, I had heard Dr. Lakkapan sir long back. We had visited your lab also, sir. Once we had around 2 days of workshop with you, so that was awesome. So I think our students will really get benefited, and all our participants who are from Pan India, they will really get benefited from your experience and expertise. So now, as I was saying, this is 6th or 7th webinar series what we are conducting, and specifically we have touched all sectors of engineering and also management. So this was one of the most unique topic which we thought, as our final years have forensic science as a paper. So we thought along with our final year students, even other participants can also get benefited with this forensic science. We would have heard a lot of times about the crime investigation, but how exactly it goes about. So to understand that, in fact, we thought organizing this forensic science webinar, sir. We are very happy and privileged to be with you, and you have dedicated your time. Now, I request Dr. Veena S. More, our Honorable Head of the Department, to welcome all the participants and the eminent speaker.
Good morning, everyone. A warm greeting from Saptagiri College of Engineering and Department of Biotechnology. Today, all of us have joined here for this webinar on forensic science in crime investigation. On this occasion, first of all, I would like to welcome our Chairman, Sri G. Dayanand sir, and Executive Director, Sri G. D. Manu sir, in their absentia. I heartily welcome today's speaker, Dr. Vinod Lakkapan, Deputy Director, Forensic Science Laboratory, Bengaluru. Welcome, Dr. Vinod.
Thank you. Thank you.
I welcome our Principal, Dr. Ramakrishna H. and heads of various departments. A warm welcome to all the participants who are students, research scholars, and faculty members from our institution and from other institutions as well. I also welcome Prashant sir, who is the convener of today's webinar. So as we all know, forensic science plays a very important role in investigating these crimes, what is happening so much in today's era. So today, Dr. Vinod is going to share his vast experiences, what he has in this field. So once again, I welcome all of you for this today's webinar. Thank you, one and all.
Thank you, madam, for providing a official welcome address for all our participants and speaker. Now, I request Dr. Soumya C., Assistant Professor, Department of Biotechnology, to introduce our eminent speaker, Dr. Vinod Lakkappan, sir. Dr. Soumya, madam.
Good morning, one and all. It's my pleasure to introduce our guest of honor and a speaker, Dr. Vinod J. Lakkappan, Deputy Director, Forensic Science Laboratory, Madhyavala, Bangalore. He has done his MSc in Biochemistry in 1997, postgraduate diploma in Computer Applications from Karnataka University, Dharwad in 2002. PhD in Biochemistry from Karnataka University, Dharwad. He worked as a senior quality control chemist with Thapar Group in 1997 to 99. He worked as a junior research fellow, Department of Biotechnology Project, PG Department of Sericulture, Karnataka University, Dharwad from 1999 to 2001. Sir worked as a senior research fellow, Swiss Agency for the Development and Cooperation SERI 2000 project. PG Department of Sericulture, Karnataka University, Dharwad from 2001 to 2004. He has acquired different positions such as scientist assistant, DNA analyst, scientific officer, assistant director, in charge deputy director in toxicological sections, Forensic Science Laboratory at Bangalore. He has presented a number of research papers in national and international conference, published his research papers in high impact international journals. He has delivered more than hundreds of invited lectures to the students and police officers, medical officers, and magistrates. Presently, he is working as a Deputy Director, Forensic Science Laboratory, Bangalore since October 2020. He's expertised in protein purifications, pigment identifications, DNA, RNA isolation, amplifications, RT-PCR, gel analysis, and primer designing and DNA profiling. Sir, I welcome you to this webinar. Thank you.
Thank you, ma'am, for the introduction. Thank you.
Sir, again, we welcome you, sir, for this webinar. Now the session is yours. So you can share your screen, sir. You are the co-host for us.
Yes, sir. Yes, sir. Thank you.
I'll just stop sharing mine, sir, so that you can share your screen.
Yeah.
Yes, sir. You can share, sir. Now new share.
Right. Are you able to see, sir?
Not yet, sir.
Just a moment.
Yes. So meantime, participants, if you have any queries, as sir is dealing with the topic forensic science in crime investigation, you can put your questions in the chat box. We'll take up the questions. Or still, if you have any questions at the end, you can raise your hand so that you can, at the end, when we have a query session, you can ask the questions. And the feedback will be shared during the last part, during the query session, and you can go for the feedback at the end, and the certificates will be delivered to your mail ID, registered mail ID.
Are you able to see, sir, the screen?
Yes, sir. So you can make it to full scape, sir.
Yeah, full screen. It was full screen.
Yes. Yes, sir. Yes, sir.
Are you able- Shall I go ahead?
Yes, sir. We are able to see, sir.
Are you able to hear me?
Yes, sir.
Yeah, thank you. Thank you so much. First of all, I thank all the faculty members of Saptagiri College of Engineering for giving me this opportunity to share whatever the little I know. It always gives me an immense pleasure whenever it comes for the students actually to tell you. Here today, what we are discussing is the forensic science in crime investigation. There are two aspects here. One is forensic science, the other one is crime investigation. So far, we have seen so many science disciplines, right? We all have gathered here to learn something which is new to the present scenario, to gain some knowledge. What is meant by the true knowledge? It is this. To know is to know that you know nothing. That is the real meaning of knowledge. So we don't know anything, take it for granted the next 1 hour that we know nothing. So let us start from the scratch. Let us try to have the best whatever I can as far as basics are concerned. We all know so far the broad disciplines of these forensic science, these sciences actually, like physical science, chemical science, and biological science. We come across these sciences many a times, but what is this forensic science? Forensic science is nothing but application of all these sciences actually. Application of physical science, application of biological science, application of chemical science, and whatnot. Sciences. That means forensic science is application of science to the law. Means in an open forum. That is a science accepted in an open forum. The open forum, you can take it as, in the current scenario, you can take it as a court. So the best open forum where you can, what you can consider. Therefore, it is also called as criminalistics. Nowadays, forensic science has become an ever-expanding branch of applied sciences. What do you think? When law comes into the picture, there are major two aspects, right? Whenever there is dispute and whenever there is a crime. What promise do you think the forensic science what we can make? The promise of forensic science, what we can make is the crime investigation. How best we can use this forensic science in the investigation of crime. That means to say effective use of forensic science is the key to better investigation. If you consider some of the important strides in forensic science is what we saw in the first picture, in the first slide, that a person holding a lens in his hands, Sherlock Holmes, right? The small magnifying lens. From the small magnifying lens, we have come across a sensitive scanning electron microscope, where you can expand and enlarge whatever the small physical evidence, you can enlarge and see the best evidence which can be linked to the crime. And then analgesics. Previously we have analgesics, now we have designer drugs. A lot of designer drugs have been designed to cure the particular disease or particular organ to deal with. Now, for the human identification is concerned, there are many techniques which have incorporated right from the blood group typing. Now we have a DNA typing where you can say, yes, you can specifically identify the individual and say, yes, there is no other than this fellow who has committed the crime. No one else is existing in this entire world. To that extent, you can prove that his involvement. And then from the calculators. We've been using calculators, small calculators, and now you can have the computers. You can do all sorts of calculations. Then the drug toxicity. Earlier, there are drugs which may have some adverse effects at some other place in our body. Now we can specifically identify with the help of toxicogenetics. We have firecrackers. Deepavali is coming up, right? We have firecrackers. Now we have high explosives. We used to identify and lift the fingerprint with the help of a fingerprint powder. Now we have a laser beam biometric system so that the person can be identified with his fingerprint. Earlier, this interrogation, lot of stuff you have to collect and then interrogate a person with their own ways. But now we have a brain fingerprinting wherein you can decode whatever that is stored in his brain, and you can easily identify that if it is his involvement is there or not. Do that. Now earlier we have manual muzzle loading guns. We have a sophisticated automatic AK-47 is currently available, and more sophisticated weapon is also available at the moment. Then we have black and white photography. You might have seen a lot of photographs earlier which are having negatives and developing these sorts of negatives to the positives. Now we have a digital photography wherein you can directly capture the images. The recorded voice, from the recorded voice to the forensic acoustics, and from the physical signatures to the digital cryptography. You have a digital signatures to do all these improvements in the forensic area in the last 100 years. Now it all starts whenever a crime scene or investigation of crime is concerned. The very basic and fundamental thing is physical evidence. This physical evidence, only thing is you need to spend a lot of time in the crime scene area where the crime has committed. You have to spend a lot of time and look for the physical evidence. And this physical evidence encompasses any and all the objects that can establish a crime has committed, and it can provide a link to its victim or the one who has committed the crime. It provides a link. Just to summarize this physical evidences, it tells us that yes, crime has been committed. Secondly, the most important thing is it links with the suspect who has committed, and it links also the links with the victim many a times, and identity of the persons involved in the crime. And it also, this is most important thing, is the one who has not committed the crime should not be punished, right? So in that case, it can exonerate the innocent. If he's really innocent, he cannot be considered as a guilty person. And then more reliable identifier, like more reliable than the eyewitnesses. The eyewitnesses who have seen the crime in their very presence during that time, and whenever it comes to the court, they can change their version because of various reasons. They may say that I have not seen any crime or anything else for various reasons. But guilt proved through science, it never changes. Right? It will be the same as it is whenever it comes to the court. And court is also having, it's all recognition in the court of law. This forensic science is having a lot of recognition in the courts of law. So that means this is most important thing. This is a crime scene. If you can consider, you have to take it as a part and look for the physical evidences, so that, which can be very crucial. Sometimes there are trace evidences. Even then you have to identify and see in the complete area whenever you happen to go to the visit to the crime scene. Now, the search for trace evidence must start from the crime scene, and it extends beyond the autopsy room. So once a crime has committed and somebody is murdered, you have to look for the trace evidences, not only at the crime scene, but at the autopsy room as well. So it can give many of the evidences which can solve the crime. And once the body is cremated, you may not get any evidences. Everything is over by then. So therefore, evidences that are, you must be very careful before the cremation of the body. And the third thing is, you must act very quickly. Like lengthy and time delay in obtaining many of the evidences, it always destroys their forensic value. No matter what sort of high-tech instruments you have in your lab, if the very evidence, if you are not properly recognized, if it has not properly been collected, and if it is not properly been preserved, then these instruments, whatever you have in your lab, are absolutely useless. Because if the very evidence goes missing, you can't do anything with your instruments, whatever you have in your lab. So this is the most important thing, is to recognize. At the crime scene, you must recognize. Once you recognize, supposing if it is a physical evidence, like a glass piece, how it is been collected or how it is been preserved. As far as preservations are concerned, the biological evidences play a role, because you must handle those evidences very carefully, and some biological evidences needs to be properly preserved. If it is not been, even if you identify, recognize, and if it is not properly been preserved, again, there will be a problem in handling when it comes to the lab. Now we have a different disciplines of forensic sciences so far, and it is ever expanding, as I said earlier, that we have anthropology, acoustics, ballistics, biology, chemical science, document, engineering, entomology, odontology, pathology, psychiatry, photography, physical sciences, and toxicology, and it goes on. In our lab, we have all these wings currently are operating, like toxicology, chemistry, biology, DNA, physics, firearms, question document, photography, forensic psychology, digital forensics, and crime scene management. We go quickly what all these divisions, exactly what is the nature of work going on in each division very quickly, and then we can focus on one particular aspect and we go in detail that particular area. As I have gone through your syllabus, biology and DNA is giving more this thing. So we'll look for how these human persons are identified with the help of forensic science. Now, when it comes to toxicology division, many of the times, almost most of the times, the answer lies in the question itself. So you must understand the question carefully. Once you understand the question carefully, you'll be able to answer it very easily. Like toxicology, the very name toxic, it says the poison. It relates to the poison. Right? It examines body fluids and organs, the presence of identification, for the presence of identification of drugs and poisons. So toxic means poison, and what is this poison is all about. The next comes poison. So if it is a poison, what it is, how it is been defined. So poison can be anything, like literally it is defined as evil, noxious, harmful, injurious, toxic, distressing, deadly, and the substance which is totally incompatible to the life. This is what the definitions, what we see when it around. But the best definition for poison, the very best definition of poison is this. Paracelsus, he gave the definition of poison as all substances, almost all substances are poisons. There is none which is not a poison. That means everything is a poison. Then how to justify this is the right dose that decides whether it's a poison or a remedy. Always the dose decides. Like if you have a fever, you go to the store and get a paracetamol. Paracetamol being an antipyretic drug, it lowers your body temperature and you'll be okay. Supposing if you take 10, 15 tablets at a time, the same remedy may act as a poison for you. That means to say all substances are poisons and the dose decides whether it is a poison or a remedy. What is this dose? The degree of toxicity of any substance, no matter whatever it may be. Its degree of toxicity, it all depends upon how much of it is entering in your body and how long it stays in your body. That is the most important thing. Its toxicity always depends on the content, the quantity, how much it enters in your body and how long it stays there in your body. This is what briefly the toxicology people they do in their lab. Now the chemistry. What is this chemistry division is doing in the lab? Deals with the cases of this adulteration of liquors. There are a lot of adulterants that are added in the liquor, and these can be identified, and also in the petroleum products. The petroleum products are adulterated. How these adulterants can be identified, there are a lot of ways to do that. Many of the equipments you can that can be easily been identified. And secondly, the narcotic drugs, other drugs that can be identified, and explosives also. This chemistry division, it deals with explosives also. These are the things, like the name suggests, chemistry. Yeah, the chemistry behind all these things, and by the application of chemical sciences, these can be identified. The next is biology deals always, name I said, biological evidences, like biological samples. They apply the knowledge of biological sciences in order to investigate biological samples, blood, body fluids, hair, fiber, and whatnot, all biological samples. Their job is to identify if it is a red stain in the crime scene. And you have to identify what it is, the red stain which is encountered. And this red stain, the very red stain, whether it is a blood or not. The first thing is whether it is a blood or not. Secondly, if it is a blood, whether it belongs to human or animal. That is the second question. If it belongs to human, what is the blood group? These are the things have been taken care by the biology people in their lab. Now, if it is a blood and if it is a group, you have to go further for the positive identification of the individual, and we'll come back to it a little later. Now, the next is the physics division. All these forensic investigations, they rely upon comparison of the evidence, right? Everywhere you are seeing the comparison. It incorporates the principles of chemistry, physics, and geology to some extent to identify and compare the physical evidence. Everywhere you can look for the comparison. Like here, these physics people, they do the compare soil, fiber, glass, and paint, and all these physical evidences. Supposing if it is a vehicle, and you have to identify, yes, this vehicle is linked to a crime scene. How will you say, yes, this vehicle is specifically linked to one particular crime scene, whether it was present at that scene or not? For this reason, what you have to do is this vehicle carries something which is there at the crime scene along with it, or else it has left something of its own in that crime scene. Supposing a glass piece of the vehicle, if it is there and you find the vehicle place, you can anytime you can have that glass piece which is found at the crime scene, and you can test the glass which is of the vehicle, and you can compare and say if it is really matching, yes, this glass piece belongs to this particular vehicle. And secondly, from the crime scene, if it takes something, maybe a soil, sometimes soil adhered to the tire, that you can take it out, the soil from the tire and the soil at the crime scene, and you can match the soil and say, yes, this soil is very much belong to the crime scene area. Majority of the times in hit and run cases, what is the exchange of paint samples. The paint samples are exchanged from one vehicle to the other. That also you can easily identify, yes, this paint belongs to this vehicle and the other one which is belongs to the other one, so that you can identify the vehicles also. That means to say you are comparing and identifying. Similarly, when it comes to the firearms, like ballistics section, it examines the firearms, discharge bullets, cartridge cases, shotgun shells, and ammunition. All these things, like in case a bullet is found in the body of the individual, that bullet is recovered from the dead body, and the vehicle is, and the weapon you have recovered at some other place. What you need to do is you have to make a test fire from the weapon, and the bullet is from the, you have to collect the bullet which is coming from the weapon and the bullet which is recovered from the dead body. You have to take these two and compare. There are specific striation marks on the bullet which is coming out from the specific weapon. This is weapon specific actually. The striation marks are weapon specific. It imparts a definite impressions on the bullet, and they are one and the same. If it is one and the same, then you can say, yes, this bullet is fired from this particular weapon. Like that, you can compare and identify. This forensic science is always a comparison and identification. Through comparison, you are identifying. The question document section. The other area is question document. Whenever the crime related to the documents, whenever you happen to identify the handwritings or the signatures, then the question document division will come handy in identifying the handwritings and the signatures, the forged signatures or the handwriting which is specific for the individual. The name itself suggests documents. Document related crimes are handled in question document division. The photography division. We have a photography division. It applies the specialized photography techniques actually, or recording of examination of physical evidences. In the crime scene, you have to take the pictures such that they can reveal many things when you take back and analyze these photographs in your lab. We have a psychology division where we can they do all these techniques like polygraph, brain mapping, and narcoanalysis, like what we say, that lie detection testing, right? Lie detection, polygraph. Through polygraph, a person can be easily be identifying that yes, he is telling lie or not. And then if something is encoded in the brain, that can be recovered through brain mapping techniques and the narcoanalysis as well. When you are introducing a chemical into the body, and then he can be in this trance state, reveal many things which is there. That means he'll be in a more talkative stage when it comes to narcoanalysis. We have a digital forensics section wherein it deals with digital devices like computers, mobile forensics. We have 3 different divisions in that: computer forensics, mobile forensics, and audio video. Whenever the audio of some person is recovered and you are individualizing it, then these audio can be individualized to a specific person if his audio voice is available, and it can be compared and matched. Other services what we have is a crime scene management unit. Whenever there is a crime and we have been called for, during that time our unit will visit the crime scene and guide the investigating officers in identifying the physical evidences and their proper collection and preservation, and to take these evidences to the laboratory for further analysis. And lastly, the DNA division. We have a DNA division wherein you can pinpoint and identify the individual, yes, specific individualization. Like in biology, as I said earlier, what they do, like whether the red stain is a blood or not, and if it is a blood, whether it belongs to human or animal. If it belongs to human, what is the blood group? These up to that are taken care of, and then for the specific identification, we go for this. With the DNA technology, you can specifically identify this individual. Now, these are the different areas, these are different sections we are operating here in our lab. Now, we'll go with the one specific area wherein you can identify related to the crime. Any crime, the main focus is on human identification. Right? For any crime, your job is to identify the criminal. Right? The best I took here, it's very difficult to explain in 1 hour, saying all the sections in quick detail. But that is why I have taken one of the areas, and I'll tell you how exactly is human identification is done through DNA technology. Prior to the DNA technology, what were the different objectives of this forensic DNA testing? The very objective of forensic DNA testing is to link, right? To link an individual to the crime scene. You are linking the individual to the crime scene or the criminal act. And secondly, the most important thing is to exonerate the suspects. And third, in case of mass disasters, your job is to identify the individuals and give away to the concerned families. When it comes to the human identification, prior to the DNA technology, so DNA technology came into the light in the year 1984-85 or so. Prior to that human identification through DNA technologies that were available for human identification are these. We have latent fingerprints through which you can identify the individual, because no two individuals have the same set of fingerprints. And then came into the light the blood grouping. To some extent, people try to identify with the help of blood grouping. And with the help of blood grouping, you can say that this individual is not involved in the crime scene if it doesn't match. But the very blood group, if it matches, you cannot say that, yes, this individual, because as you all know that we all have different, same type of blood groups in quite huge number of population. And then protein polymorphism. We all have different sorts of proteins in our body, right? Through which the individuals can be identified through protein polymorphism. It's a little difficult, but it's possible. And then skull superimposition. Earlier, prior to the DNA technology, we do have this skull superimposition, wherein you have a skull of the dead body, and if you have his life-size photograph, you can superimpose his face with the skull and identify to some extent, like 75 to 80%, you can identify, yes, this skull belongs to this particular individual, but you cannot say specifically with this technology. When the DNA technology came into the light, now we start doing this skull superimposition technology method. We'll see one by one how it works. Like latent fingerprints, as in the year 1860, William Herschel, to identify with the help of latent fingerprints, what he did was he took his own prints in the year 1860. Then after 28 years, he took the prints one more time. Now he matches with the one which he took earlier. He did not find any difference at all. What is the conclusion? That the fingerprints do not change over a period of time. Right? And later he has shown us that no two individuals have the same set of fingerprints. So wherever these prints are available in the crime scene, we have a separate fingerprint bureau unit. These people, they go and collect these fingerprints from the crime scene, and then they try to match with the individuals who are suspects. Now the secondly, the blood grouping. The Indian population, what it says is 40% of the Indian population is of O group. You can see here in the end, the blood group O, it is 40. Like 40% of we people are of O group. In that case, it is not possible to identify specifically that the individual who has committed the crime. Now, the final word on identification today, what we have is DNA fingerprinting, through which you can specifically identify the individual. This DNA fingerprint, unlike conventional fingerprints that are outside your body, this DNA fingerprint, it lies inside your body because each of your cell has got this information. So that is the reason why we say, yes, DNA fingerprint lies inside your body. Nowadays, people are rarely calling it as a DNA fingerprint, but the more sophisticated word is DNA profiling or DNA typing. So don't get confused when I use DNA typing or DNA profiling. It is nothing but DNA fingerprinting only. If you want me to explain what is this DNA fingerprinting is all about in a single sentence, method of identifying an individual by the unique characteristics of that individual's DNA. That means each one of us is having the DNA, and this DNA has got the unique characteristics and through which you can identify the individual. This is a method of identification of the individuals. You all know this. What is that which is present in DNA is all these 4 chemicals. Other than that, you can't find anything like adenine, guanine, cytosine, and thymine. You all know this, right? The very basic thing is for the identification is we all have 50% of DNA from our parents. That is the most important thing here to look for. We have 50% of DNA from our father and 50% of DNA from our mother. Again, through comparison, you are identifying the individual. Whenever I say DNA fingerprinting, the 2 things should go straight away into your mind are what it does, DNA fingerprinting, identification, right? It is used for identification purpose, and how this identification is done is through comparison, like. So that is the reason why I say DNA fingerprinting, whenever we come across, the 2 things are, one is identification and secondly, comparison. So where you find this DNA inside your body is this. If you take it as a cell, there are a lot of components in the cell. Where you find DNA is inside the nucleus. Right? If you take out the nucleus from the cell, it's like this. If you take out the nucleus, you find the DNA and as well as the proteins also. The black, white dotted structures in between look like what is histone proteins, what we. So the chromosomes are made up of the DNA and histone proteins. So all living things, like every living thing is made up of cells, and these cells contain the nucleus. And inside the nucleus, you find the chromosomes. And on the chromosomes, you find the DNA. It's as simple as that. What is the function of DNA then? The function of DNA, like practically speaking, each function of your body is controlled by the DNA. What we say is a nucleus is a control center, right? The nucleus, as the nucleus is a center and you find the DNA inside the nucleus, that is the reason why we say that each of your function is controlled by DNA. That is the reason it is called as a blueprint of life as well. It is called as a code. Why it is called as a code? Because it is all having these 4 letters like A, G, C, T's. It's a double standard structure. You can see below the letters G, G, A, AAC, right? The letters which are there on the stand, I have written it down. And they are all like in AGCTs only. If I ask somebody it as a word, it is not possible for anyone to read it as a single word. Like if I write STANDARD, that a standard one, but you cannot read it as a word. That is the reason why we call it as a code. It is, we do not know what it is, but it has got its own meaning, like saying what exactly, like which it has to be, it is coding, and all these coding details will be there in the code, and we may not be able to identify it. That is the reason why we are calling it as a code. DNA fingerprinting is one of the most conventional methods of human identification. As far as human identification is concerned, a DNA fingerprint comes at the top. And it is more reliable identifier. Why? Because if you do the testing here in Bangalore or at your place or in Delhi, Mumbai, England, America, wherever, if you do the testing, the results are going to be the same. As far as the samples are same, the results are going to be the same. And thirdly, there are so many ways to get a print. Unlike conventional prints, wherein if you go to the crime scene, if you do not find the fingerprints, conventional fingerprints, it is not possible to identify that individual through conventional fingerprints. But when it comes to the DNA fingerprinting, all you need is a biological sample, whatever it may be. All you need is a small biological sample that you must look for whenever you visit the crime scene. It may be a small speck of blood, a used cigarette butt, wherein you can have the saliva sample, semen, skin, tissue, teeth, or 1 or 2 hairs in the crime scene. If you are identified, then that can be easily collected and the person can be identified through this. There are so many ways to get a print, because any biological sample, it is not that you have to get the blood all the time. No, any biological sample, because all these made up of cells containing the DNA. The important main 4 basic things whenever it comes to the characteristics of DNA is that each person has a unique DNA fingerprint. Like each individual's DNA is the same in all the cells. An individual's DNA profile remains same throughout his life and even after his death also. And as I said earlier, half of the DNA comes from each of a parent, like 50% DNA comes from 50% from the mother. Like William Herschel in the 1860s, he has shown us for the first time how this conventional fingerprinting he has talked about. In the same, we have DNA fingerprinting, like Sir Alec Jeffreys in the year 1985, he has shown us for the first time how this DNA fingerprinting is done. Again, it is a comparison, as I said, identification through comparison. Comparison of DNA from 2 different sources. If you find only one, like supposing you're finding a bone sample at some other crime place, and if you take it to the lab and if at all you need to identify, it is not possible. It is not possible and it cannot be individualized to a single person. Why? Because no, it will give you a DNA profile. Take out the DNA from the bone and you can subject it to the extraction properly. No doubt, it will give you a DNA profile for you. But the person cannot be identified because there is no sample for comparison. So how this bone can be identified is either you have to have his own sample which is collected earlier, or you need to have the samples of his or her parents to compare and match and conclude. The chemical structure of everyone's DNA is same. Like we all have the DNA and its chemical structure is same. The only difference is the base pairs. That is the reason why we are all different. Why? We all have the DNA. Why we are not same? Why we are all different? Like you all have your mobile phones, right? And these mobile phones are made up of mobile phone numbers 0, 1, 2, 3, 4, 5, 6, 7, 8, 9. If I pick up my phone and dial 0, 1, 2, 3, 4, 5, 6, 7, 8, 9 and dial, it has to give ring to each one of your mobiles. But does it so? No. Why? Because the sequence of this number is different. Like these mobile phone number digits are different. Likewise, 3.3 billion letters, if they're arranged in different permutations and combinations, you can say no two individuals are the same. Each one of you is unique. So there are so many millions of base pairs in each one of your individual's DNA, and that every person has got a different sequence. Whenever I talk of DNA technology, there are two important DNA technologies. One is what we are discussing right now here is DNA fingerprinting. The other one is DNA sequencing. Now you all know what is DNA fingerprinting. It's a method of identifying an individual with the help of unique characteristics of that individual's DNA. That is what is all about, this DNA fingerprinting. What is DNA sequencing? I told you earlier also that if you look at the question carefully, the answer lies in it. A sequence. Sequence is arrangement of the letters, right? Arrangement of AGCTs. If I ask you what is the sequence of your mobile number, and you are able to tell that what is the mobile number, that is the sequence of your mobile number. Likewise, DNA sequencing means the arrangement of all these 3.3 billion letters, how they are arranged. That is DNA sequencing. Arrangement of base pairs. Now, just to refresh your memory, so this I have taken it from your second year PUC books, just to make your basics strong. As simple as that, you all have studied this. What it says is 99% of DNA does not differ from one another. Right? 99%. The difference is hardly 1%. Actually speaking, it is even less than that, 0.001%. It is very, very less than 1%. Now, whenever you are identifying somebody, what is that you are looking for? Are you looking for the similarities, or you look for variations? Always people look for variations, like what is the difference between these two? Look for the variation, not similarities. Here also, even if it is 99% of DNA is common, we look for variations. If it is less than 1% also, you can easily identify. Like I normally used to give an example whenever I talk of this slide. Like you have invited some 10 different individuals in your college, and you don't know anything of any individual. You don't know anything of anybody. All of them, they are there on the dais. They are all wearing white shirts and black pants and brown shoe and a blue cap on their head. That means 99% common. And they are wearing different colored ties. If at all you want to identify, to talk of somebody to your next person sitting next to you, what is that you are, is a tie color, not the one which is similar. You are looking for the variation. Here the variation is a tie color, right? In that case, we are looking for the variations, not the similarities. And these variations across. Now this human genome has about 3 billion base pairs, like 3.3 billion cells, base pairs, bases. And out of which 5% of this DNA functions as genes. That means 5% which is coding for the proteins. And the remaining 95% of DNA, we do not know what it does till today. We call it as junk DNA, which is of absolutely no use. Junk, we do not know its function. That is the reason why we are calling it as a junk, because we do not know. The interesting point here, the variations, what you are looking for, you find it in the junk DNA. This junk DNA portions that contains some repeating sequences of base pairs. And these repeating sequences, hope you remember, you refresh your memory, it is called as variable number tandem repeats. You might have heard this VNTRs, right? Variable number tandem repeats. These are the variations which is found in. And each VNTR, it may contain 20 to 100 base pairs. A VNTR is a definite sequential repeat. Particular sequence, if it gets repeated more than 30 times in a row, 100 times it can repeat. And each individual has a unique pattern of these VNTRs. Between parent and child, 50% of similarity is there. You can find it in VNTR. A repeating unit, that means it has got a continuous repeat. Supposing, just for you to give an example, if I take the repeat letters as STANDARD, suppose consider it as a VNTR, STANDARD, and it has to repeat continuously, like standard, 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 standard. Repeatedly, it has to repeat. Then you can consider, yes, this is a VNTR because it has got number of repeats, the same repeats. Only then you can take it as a repeat. Now the DNA profiling, it all depends upon the junk DNA, as I said. Junk DNA is also called as non-coding DNA. You know that 5% of DNA which codes for the protein, and we call it as a coding DNA or functional genes. Like the other 95% which does not code for any proteins, which is called as non-coding DNA or a junk DNA. And why we are looking for this junk DNA? Because we look for variations and we find these variations in the junk DNA. So that is the reason why we are on the junk DNA, because through which you can identify, and our job is to identify the individual. And the modern profiling, presently we are doing it as short tangents. The modern technology, STRs. Earlier it was VNTRs and STRs, and the name itself clarifies that short tandem. The repeat number is very small, usually 2 to 5 bases of length. If you consider as 2 to 5 bases, they are in length and they are continuously repeating, then you can take it as STR. Only thing is, it has to have the continuous repeats. Unlike VNTR, where you can find 20 or more bases which are in continuous repeats, likewise you have STRs, which is short tandem repeat, 2 to 5 bases in length. If you do this DNA sequencing of one particular individual, it look like this. Like 3.3 billion letters, how they are arranged. This is what we call it as DNA sequencing. But it doesn't help us in identifying the individual. It can give you the sequence to study for further areas of further aspects of DNA if it is required. What you need to look for is, are there any continuous repeats? Any few bases which are repeating? You have to look for these repeats. If you look at carefully this slide, you can find some bases are repeating continuously. Make out what are those repeats. I'll just make your job a little more easier. See here. Now you are able to identify, right? Yeah. What is that repeat called? GG80. That means your DNA, in your DNA, you have a GG80 repeat. If at all you want to identify this GG80 repeat, what are the 4 bases you need? The complementary bases you have studied, right? What is the complementary base for G? the complementary base for G is C, right? The first one is C, then again G. For G, you have C. For A, you have T, and for T, you have A. That means to say, if you use CCTA, it acts as an STR marker for you. Use CCTA, it identifies GGAT, and you have to look for the continuous repeats. You can see in the end, there are a lot of continuous repeats. If you consider this as a location, like from base 1 to base 1000, each one of us has got these repeats. In a human population, each one of us has got this sequence, these repeats, and the only difference is we have different repeat numbers. I may have 10 continuous repeats, you may have 12, and somebody else may have 14 or 25 or 24, like that. But the thing is, each one of us has got this repeat, GGAT. Likewise, we have 16 different locations wherein you can identify 16 markers. Now, how this DNA analysis is done? The process is very simple. You must require 2 samples. As I said, with a single sample, it is not possible to identify specifically that individual. You have to look for the biological sample at the crime scene, collect it, preserve it if it is necessary, and then take it to the lab. And the police people, they got hold of some suspects. If there are 10 different suspects, they collect the samples of the biology, like blood or whatever may be the sample. And these samples will come to your lab, and you are doing the DNA extractions and analysis, and you're identifying it through comparison. The biological sample which is collected at the crime scene, and the biological sample which is given to you as a suspect. So these two are compared, and if it matches, you can say, yes, it matches. And if it doesn't match, you can say it's not matching. That means to say DNA fingerprinting is a comparative process. DNA from crime scene is compared with the suspect, and therefore a minimum 2 samples must be required. Currently, there are 2 methods as far as DNA fingerprinting is concerned. The one which gave Sir Alec Jeffrey gave us in the year 1980, which depends upon the VNTRs, and the method is called as RFLP, restriction fragment length polymorphism. This you have read it, and it is there very much in all the textbooks. What you have, this method, restriction fragment length polymorphism. And now the latest method, all labs have adopted this, the PCR based method called as STR typing, short tandem repeats. That means there are only 2 methods. One is VNTR based, the other one is STR based. Any method, if you can take any method, the first job is to have a biological sample, right? You have the biological sample to start with. It can be anything like blood, saliva, semen, skin, bone, hair, teeth, or whatever. It all starts with the biological samples. And then it goes for a lot of processes in the, if you consider as the restriction fragment length polymorphism, there are a lot of procedures are involved, right from the DNA extraction, quantification, how much of DNA is there in that. And then if it is more DNA, you need not have to go for a PCR for amplification in VNTR method. And then you have to denature it, double stranded to single stranded, then southern blotting and all these things. And in the end, you get on an X-ray film with autoradiography, you have this picture on your X-ray sheet. Started with the 3 different individuals, then all the 3 patterns are different. If you start with the different biological samples of the same individual, then you can expect the same banding pattern on the three, because all these samples belonging to the same individual. This RFLP method is very time consuming. Like if you start today and it gets completed only after 10-15 days, it requires a lot of time and then laborious. A lot of processes are involved, and it requires DNA of good quality, and it requires more amount of DNA as well. So that is the reason why people are stopped doing it, because you have to have the results as quickly as possible so that the guilty can be punished earlier. The new technology, that is the reason why the new technology we have here, like STR analysis, it has largely replaced the original RFLP method, DNA fingerprinting, and everyone, all of the labs are adopted this same STR technology. Here, a small, if you have a type of blood sample or any other biological sample, is more than sufficient to identify very small speck, like a nanogram of material biological sample is enough to identify through DNA technology by STR method. So what is this STRs are all about? This is the genetic analyzer machine, wherein in RFLP you are doing the gel running manually. Here the machine does with the capillary. The principle is going to be the same. The fragments, they are net negatively charged and move towards the positive electrode, and shorter fragment moves faster. It has all been taken care by the genetic analyzer. So this is the machine, how it looked like, the older machine. When you open the doors of the machine, it looked like this. A capillary electrode you can see, and at the bottom you can see there are a lot of samples placed for provision to keep more than 90 samples at once. And it picks up each sample at a time, because there is only one electrode and one capillary. It picks up one sample and it goes to the detector, and detector detects how many fragments are there, and it gives you as a profile. Now this is the next version of the machine, wherein you can find 8 different electrodes and 16 different electrodes, 8 one behind the other, and 16 different capillaries. That means you can do 16 samples at once. One sample is taking 40 minutes, somewhere around 40 minutes to 1 hour. That means here with this machine, you can do 16 samples in 1 hour. If I zoom it, it looks like this. Here you can see the electrodes very clearly, like 8 and 8, 16 electrodes and 16 capillaries. After the completion of the DNA profiling, it looked like this. All the banding patterns, like it looked like a barcode. If you go to the supermarket, all the items, they have the barcodes and they look like this. That is the reason why initially it called as a DNA, it called as a fingerprint, a barcode sort of a thing. All these samples are giving different coding patterns. And at the same time, it is giving you a DNA profile as well. This is the DNA profile. Wherever you find peaks, then you call it as a profile. Here, all the labs, they have using 16 different set of markers, 16 different locations. Like why 16? Because see, world population is much more than 10 to the power of 16. If the world population goes increasing, and if it is more than 10 to the power of 16, then you need to have a few more number of STR markers, so that you can say that no two individuals have the same set of DNA profile. Now it is very much less than 10 to the power of 16. You can these 16 markers are more than sufficient to say that no two individuals have the same set of DNA profile. Currently, there are a lot of many companies, they have come up with the markers like 23 markers, 25 and 7 markers currently available and can be used for high power of discrimination. In a banding pattern, it is very difficult to identify the bands when it is a little faint band. When the sample is limited, you may not identify if it is very faint. But here, you can easily see that a number below the peak, number below each of the peak, you can see that 12, 16, 29, 30, and so on. And if you look at the last in the red peaks, you can see X and Y. That means that DNA profile belongs to a male individual. If it is a female, you can see XX, a single peak which is showing XX. It's a female individual. Like each of the section, you find two different peaks. That means 50% of the DNA from the mother, 50 from the father. Like 12 from the mother and 16 from the father. Like 50% of the mother, 50% father. In all cases, you can see the two different peaks. Even though you may see a single peak at some place, but you can see the numbers below. It is giving you the same numbers twice. Like in first row, it is 12, 12. Second also, it is a green peak, which is a single one, which is showing 12, 12. And in third, you can see that then there are two areas where you can see a single like 17, 17 and 8, 8. Like means same number of repeats. In a population, there are every chances that few of the locations may have the same set of number of repeats. What these represent is the number of repeats, whatever we are looking for. That GG80 I was talking about earlier, like this GG80 repeated 12 times and in the father's this thing and 16 times in the mother at the same location. Likewise, there are different repeats. What you need to identify is through sequencing, you must know that what is the repeat unit. And once you know the repeat, what are the repeats are there in that particular, you can easily design an STR through base pairing technology. You know that whenever there is G, it is pairing with C, and whenever there is A, it is pairing with D. There are a lot of advantages when it comes to DNA fingerprinting. One is PCR based. This STR method is PCR based. Without PCR, it is not possible to perform STR. But without PCR, it is possible to VNTR technology like RFLP method is possible. If the DNA is more PCR. Without PCR, you can perform the DNA fingerprinting. And if it is a low quality DNA also, you can do the DNA testing. And if it is a degraded DNA, that also possible. So we have 2-3 years ago, we have heard about there are a lot of skulls recovered at one particular area near Dharwad. A lot of skulls have been recovered. And when it comes to identification, the samples for comparison was not available. As I said, if it is a single sample, it is not possible to identify and to one specific person, but no doubt it gives the DNA profile, but it is not possible to identify through single sample. Highly discriminating. There are a lot of other locations are also available, and it's abundant in genome. A lot of other locations wherever you can find a different set of repeats are available, and rapid typing. Through STR technology, it is possible to do the DNA fingerprinting within 24 hours, unlike RFLP method, wherein you have you require hardly more than 15 days. Now here, it's a rapid, as you can say, you can do the DNA fingerprinting in less than 24 hours. And it is a non-isotopic, radioactive isotope RFLP method. Here it is a non-isotopic method and discrete alleles. That means you see the numbers very carefully in each of the DNA profile. You get the numbers, not the bands. You get the bands as well, but you are getting a DNA profile as well, wherein you can easily identify the numbers and tabulate it and identify. And amenable to automation. There are more of the machine which takes care of everything. It's not like manually when it comes for RFLP method, there are more manual methods what you need to do. And here you can have the machines which takes care of everything. And last, in the end, to look for some of the applications we'll see, and then we'll wind it up. Lot of applications are there when it for the DNA fingerprinting. You can do paternity, maternity disputes. You can solve the rape and murder cases, and the baby exchange in hospitals. There are quite a few cases wherein we have encountered like baby exchanges in hospitals and mass disasters. A lot of incidents in mass disasters. It's not a crime, but when it comes to disasters, it is your job to identify the individuals and hand over the dead bodies to the concerned relatives. And then identification of mutilated remains, authentication of plant seeds as well, quite a few applications, forensic wildlife, huge number of applications. Other applications are in plant biotechnology. You have biodiversity, immigration, people are coming from outside the country. Inheritance matters also, you have huge number of applications. There are types of cases, as I said, paternity and maternity disputes, and samples in rape and murder cases. Identity, like bones and teeth or whatever, the skulls, these type of cases. And I just wanted to discuss quite a few simple cases with this. If at all you have identified and you have learned so far, if you have understood it carefully, you must be in a position to answer my questions, whatever I'm putting before you. So these are the, say, normally in a case where you in a paternity disputes, 3 different blood samples, like one belongs to the mother, the other one is child and the father. Now your job is to identify whether this child belongs to his parents or not. So this is the banding pattern through which you can identify. There are different locations here. And in the middle, you have the child's banding pattern. All you need to do is you look for one band is there in the father's profile or not, and the other one is there in the mother's. That is the only thing what you can compare. In a banding pattern, as I said earlier, some of the bands are little faint. You may not be in a position to identify, but when it comes to a profile, it is very much clear that it all gives you the numbers like 15, 17, 8, 9, 9, 14 and all these numbers. It is very easy for you to tabulate and identify and compare. Suppose this is a picture of a DNA gel. On the gel, the first one is standard, second one is the mother's DNA profile, third is the baby, and the fourth is the father. With respect to one marker, and a diagram with one particular marker, can anybody tell me that whether this baby belongs to parents or not? Okay, what you need to do is in the third row, the first one band, which is there, fourth, that means in the father's DNA, which has come from Now you can compare and you can easily that whether this child belongs to these parents or not. Always DNA profile in between, because what you need to do is in each of the locus, first row, one number should be there in the mother's profile and the other number should be there in the father's profile if this child belongs to these parents. Just look at this slide and see the numbers are matching or not. I'll make your job a little more easier now. You can see here in the middle child's DNA profile, all the black ones from the mother and all the blue numbers, they are coming from the father. That means 50-50. The concept is 50-50. We all have 50% of the DNA from our parents, like 50% from the father and 50% from the mother. In the next case, here also you get 3 different samples from the mother, disputed child, and the father. You can see this A, B, C, D, wherever I mark the yellow one, A, in the second row, second column, like in the child's DNA fingerprinting, that yellow one is not there in the either in mother or father. Likewise, in the B, you have a blue one. In the C, again, there is a blue, and in the D, there is a green colored band. But if it comes to a number, it is very easy to identify. Now you tell me whether this child belongs to these parents or not. First, you look for the mother, because there are every chances that in a hospital, if the very mother, if the baby is exchanged, the baby's profile is not matching with the mother. So that is the reason why you need to look for the mother's profile and it matches or not. One number is coming from the mother or not, the child's DNA profile. If you can see here, yeah. See in the first, it is 14 is there, then 28, 7, all the mothers, black ones, which is there here. But some of the numbers, they are not matching with the father's DNA profile. Like the first one, 15, which is not there in the father's profile. Likewise, in the second, 33.2, it is not there in the father's. If quite a few numbers doesn't match, you can say that, yes, he is not the biological father of that individual. So if it is a baby, suppose consider this a baby exchange case wherein the babies are handed over to the wrong parents initially. The first one is baby 1, baby 2. Then what you need is the parent sample of the babies, like father, mother of the first baby and father, mother of the second baby. All you need is a 6 samples. Then you can compare the baby 1 with the parent sample. See here. Now the baby 1 is matching with the father 2 and mother 2, and the baby 2, all the numbers are matching with father 1 and mother 1. So this is how you can identify through DNA technology. So it's a case of a murder wherein a tissue sample is recovered from the dead body, and a small speck of blood on the cloth shirt of somebody, which is recovered later, and these two have come to your lab and you are doing the DNA fingerprinting. It says, yes, this biological sample which is found on the shirt is very much is that of the deceased. So this is how you can identify straight away that. Here again, there is a 3 different suspects, like A, B, C are the suspects and F is the forensic sample. If you consider it a forensic sample as F, then what you need to do is to compare the banding pattern which is there in the F, which is matching with which number, which individual. See here, even though it is matching with A and B, the first band in the F, it is matching with A and B. The second one is also matching with A and B. And the third one in the F, third band, which is matching with only B, right? Then all the other remaining are matching with B. Now, yes, the involvement of B in the forensic question sample. At the same time, you can say that other two, they have no role to play in this, because as the very sample as the sample of A and C are not matching, that is also equally important, that which is not matching also you can specifically say. That is another advantage of this DNA technology. Now the identification of mutilated remains. Once the person dies, what you can expect is the postmortem blood sample, bone marrow, skin, tissue, teeth, hair, and all these samples. Even after the death, whatever the sample biological available, you can identify and specifically identify. This is a picture of 20 years back when the World Trade Center collapsed. 3,000 people have died. And in that place, there are more than 22,000 tissue pieces. And what you need to do is to do the DNA sample extraction and profiling of all these 22,000 tissue pieces, and then bring it together, which is giving the same DNA profile, because all these biological samples belong to that individual, and then you have to match it with the parent sample. That there was a database like CODIS, what we call it as combined. I'll come back to it a little later. There was a database wherein in that database, their DNA profile, which is stored earlier, and majority of the dead bodies have been identified through this database, because you need not have to have the parent sample for comparison. You have the database, do the DNA extraction of the sample which is found at the place, and get the DNA profile and match it with the database. If it gives the match, it straight away tells us to whose sample it is. In that way, it is identified more number of dead bodies. Here is an accident which took place 3 years ago, 7 years ago, I believe. Bangalore Nanded train accident. An AC bogie caught fire. More than 27 people have been charred to death, and their bodies have been in a state of beyond recognition, and through DNA technology, it has been identified. And earlier cases also, in a case of late former Prime Minister Rajiv Gandhi assassination case, this DNA technology helped us in identifying. And Nana Sani Tandur murder case, her own husband's involvement. And then Madhumita's case also, Karnataka's first homicidal case, Beena murder case also, which is solved with this DNA technology. And if you take it as an international case, with the first case which solved with the DNA technology is this. In England in the year 1983, a 15-year-old girl was raped and murdered in the year 1983. And after 3 years later, in 1986, the second murder took place. And one fellow, he came forward, his name was Richard Buckland, and he confessed, yes, I did both the two murders. But the DNA test showed that the same man killed both the girls, but it was not Buckland who has come forward and confessed. He was not the person. Then the police people, they asked to give the sample of all the villagers. There were about 5,500 people who were there in the village, to give their DNA. All of them they gave except one person who did not give the sample for DNA testing. His name was Colin Pitchfork, and he was the person whose DNA which got matched with the question sample. And this was the database I was mentioning earlier, a combined DNA index system. We do not have it here in India, this database, but it gives a lot of information saying that if you have the DNA, only one sample, you can pick up the biological sample at the crime scene, DNA testing in your lab, get the DNA profile and match it with the database. You need not have to trouble all other suspects who are not involved. But there are certain issues also why it is getting so much of time to get it approved. Many of the countries, they have the database of this so that they can be used for identification purpose. last two slides. I'll just end the last two slides. So this is nothing to do with the DNA fingerprinting technology. Just to give your information, DNA from a single human cell. If you take the single human cell and pull it as a thread, it will be somewhere around 2 meters long. A single human cell DNA, and it contains the information of more than 600,000 printed pages, and each page is having somewhere around 500. That means you're all a mobile library of more than 1,000 books of information. As I said, each cell has got somewhere around 2 meters long of DNA, and an average human being has got more than 3 trillion cells. If you take out the DNA from 3 billion cells, if you take out the DNA of one individual, and if you pull it as a thread, the distance would be from Earth to Sun more than 400 times the distance between these two. Such a long thread, and it has got the diameter of 2 nanometers of diameter. Let me stop here. I think I took more of the time. Hello?
Yes, sir.
Yeah, let me stop it here. And if you have any queries, you please.
Okay, participants, now we'll have a Q&A. If any questions, please you can ask. We'll unmute, you can put your hands up, or you can unmute and then you can start asking. Or you can put your questions in the chat box.
Hello. Prashant,
Yeah.
Hello.
Yes, sir. Deshpande, sir.
Yes, sir. Sir, that was a wonderful presentation. Created a lot of interest and curiosity in this subject. And I have two questions, sir. One is, if our students are interested to enter into this area, what should be the minimum qualification, or what are the courses they may have to undergo to enter into this field? That is one question. Second is, even though you have shown so much of improvements in technology, so why that delay is happening in settling the cases? That's another question. Doubt I have.
One?
No.
Which
you in a crime scene, even though the technology has advanced so much, some cases we were not able to solve. So that just I wanted to know why that is happening.
Yeah. Let me answer your first question. How you can enter into the forensic field. Currently, as per the CNR rule, what it says is the entry stage is MSc plus 2 years of research experience. As you can enter it as a scientific officer. That was the initial entry for the post. Scientific officer, you can enter with the qualification of MSc plus 2 years of research experience. That is what currently it says. And secondly, there are a lot of crimes which have gone unsolved. Very true. I totally agree. The thing is, it is not possible to attend each and every crime scene, to go and attend at that place for a forensic expert. It's not possible. It is true that we can attend quite a few number, but not all. It is practically not possible. That too, in Bangalore, we can reach. In other places, it is a little difficult. And the second part is that, see, people go wrong, or they may not identify the specific evidence or specific clue material as a physical evidence mentioned earlier, saying that the very clue, the very physical evidence, if it goes missing, that if you can't recognize it, no matter what we have the machines in our lab, we can't do any miracles in our lab if the very evidence goes missing, which we fail to identify at the crime scene.
Okay, sir. Thank you so much for the information. Thank you.
Yes, sir.
Participants, you can unmute. Thanks, Raghavendra Deshpande, sir. Sir, he is our professor from mechanical engineering. Lot of interest with biotechnology, in fact, sir has got. So for every webinar, sir participates very actively, sir.
That's great, sir. That's great.
Students, any questions? You can unmute yourself as Deshpande sir did, and you can ask questions, clarify your doubts.
Hello, sir.
Yeah.
Good evening, sir. Good morning, sir. It was a very good presentation.
Morning.
Any private labs are doing this DNA fingerprinting, sir?
Actually speaking, private labs are there, but I don't think they are doing it here. They are collecting the samples and sending it out.
Okay, sir. Okay.
As a mediator,
Okay.
they are working as a mediator. They are collecting the samples here and sending it to the lab like CDFD, Hyderabad, or any other labs who are doing.
Okay.
And they collect the report from there and give it to the persons.
Which is the ideal sample if you want to do the test, sir?
Ideal sample, normally they collect the blood sample.
Okay.
Because, see, blood sample, you can easily collect it and you can store it on a paper, like what we call it as a FTA paper, wherein you can put a drop of blood on that paper, and if you dry it for quite some time, like 1 or 2 hours in a shade, it can stay for long. It can stay for many years. Even after 10-15 years, you can take out that paper and you can store it at room temperature. No need to have be preserved at colder temperatures. Even if you store it at room temperature, you can take out that sample after 15-20 years also, you can get the DNA profile out of that. That is the second reason why sample is the best.
Thank you very much, sir.
Thank you, Kumar, sir. There is a question at the chat box. What is the most challenging part of this job? A student has asked.
Yeah, the most.
Student by name Amruta has asked, sir.
Yeah, it's a good question, actually. I really appreciate. What is the challenging? Like, in any case, see, the challenging thing is the identification of the clue material at the crime scene. That is a real big challenge. Once it is identified, and then the next part is the extraction part. If it consists to the DNA fingerprinting, the first thing is extraction part. If you do the good extraction of DNA, the rest of the things will easily follow. If you go wrong in doing the extraction, saying that you are extracting the DNA, and if you spill out the DNA and if you have the protein, it may not give you the DNA profile. So the extraction part, identification of the clue material, identification of the biological sample at the crime scene, and the DNA extraction plays a vital role. It's as good as a challenge, let me tell you, because it's not that easy as I've shown here, because taking out of DNA from the blood and other samples is quite easy, but when it comes to the bones and teeth, it's really a tough task for anybody to get the DNA out of it, because a lot of processes are involved when it comes to bones and teeth. There are a lot of procedures are involved, and if you do not get the DNA profile in the end, you cannot go back to the previous step and redo the things. No, you have to go back to the first step and redo the entire exercise. So that is the reason why I'm telling you the extraction part is really vital. A very good question.
Thank you, sir. Any other questions? Right, sir. I think we have come to the end. Now I request Professor Ramya DL, Assistant Professor from Department of Biotech, to propose vote of thanks.
Good afternoon, everyone. Myself, Professor Ramya, Assistant Professor, Department of Biotechnology, Saptagiri College of Engineering. I take this privilege in proposing vote of thanks. First, I would like to thank today's webinar speaker, Dr. Vinod Lakkapan, our Deputy Director, Forensic Science Lab, for his valuable interaction and expertise sharing. I would like to thank you, sir. And next, I would like to thank for the organization, and please accept our appreciation certificate here. Once again, thank you, sir. I also thank our chairperson, Dr. G. Dayanand, and our executive director, Shri G.D. Manoj sir, for their continuous support. Now, I thank our principal, Dr. Ramakrishna H., for his encouragement in conducting of such events. Now, I extend my thanks to our HOD, Dr. Veena S. More, for her continuous support. I thank all the participants from Karnataka and all our India who have witnessed such an informative webinar at Zoom platform and YouTube Live. Last but not least, I would like to thank Dr. Ravi sir, HOD, Electrical Department, and Professor Yusuf for timely providing technical help in organizing this webinar in a short duration. And I also thank this webinar convener, Professor Prashant Kumar HP, for his interest, time, and commitment towards conducting this kind of events in our department. I also extend my thanks to the coordinators and staff of Biotechnology Department who directly or indirectly involved in organizing such successful webinar. Okay, thank you all.
Thank you, sir, for your time in the busy schedule. Vinod, sir, it was indeed very informative. You have covered the entire, all the basic services, as well as you have covered most of the, with the case studies, most of the things. So our students would have got really benefited. So thanks a lot, sir, for your time with the busy schedule. Once again, I thank you, sir. For the participants, we have already put up feedback in the chat box. Please provide the feedback, and the registered, whatever the registered mail ID you will have, you will get your e-certificates. Thank you, sir. Once again, it was indeed very happy. And for all participants, thank you. Thanks a lot. We will wait for another 10 minutes.
Thank you. Thank you.
Chat box. In the chat box, we'll have that feedback link for another 10 to 15 minutes. Thank you, Vinod, sir, for making our day.
Thank you, sir. Yeah, thank you, sir. Thank you so much. It always gives a pleasure whenever it comes for the students actually to tell you. Thank you very much.
Absolutely.
Thank you.
Thank you, sir. Bye, sir. Participants, even we have posted the feedback link in the WhatsApp group, which has been created. You can also check over there. Thank you. Thanks a lot.
Thank you very much, sir.
ನಾನು ಸ್ಕ್ರೀನ್ ಶಾಟ್ ಅವಾಗಲೇ ಅನ್ಕೊಂಡೆ
ನೋಡಿಲ್ಲ.
ನೆಟ್ವರ್ಕ್ ನಾನು
ಫೋಟೋ ಚೆನ್ನಾಗಿದೆ.
Participants who have already completed with the feedback, I request you can leave the meeting, and even feedback is also shared in the WhatsApp group in which you are there. So you can take from there also. Thank you. Rameshana?